welcome to our lecture on touch and pain, in which we will briefly discuss the somatosensory system, and then we will focus on how people perceive pain. So we cannot begin our lecture without first looking at skin anatomy. The skin is divided into several layers: the epidermis, the dermis, and the hypodermis. And there are certain receptors embedded within the layers of our skin that respond to different types of stimulation, such as bending and stretching. And these are called mechanoreceptors. They, as we see, are different types, and they are located throughout the layers of our skin. We also have free nerve endings, and these are the ones that enable pain perception. Now, the mechanoreceptors are numerous, and they tend to preferentially pick on a variety of stimulus frequencies, pressures, and they have varying receptive field sizes and also adaptation rates. Now, what happens when we stimulate one patch of skin? Let's say we touch something with the tip of a finger. Well, this triggers sensory transduction, which is the process of converting the energy of a stimulus into an electrical signal. In turn, this triggers receptor potential, which is proportional to the intensity of the stimulus. Subsequently, this receptor potential triggers action potentials, which are generated in afferent fibers whose cell bodies are in the dorsal root ganglia of our spinal cord. Most of these afferent fiber terminals that detect and transmit tactile sensory stimuli are encapsulated by specialized receptor cells that help tune this afferent fiber to somatic stimulation. And these encapsulated fiber terminals usually have lower thresholds for generating action potentials, meaning that they are more sensitive to sensory stimulation compared to free nerve endings. On the other hand, unencapsulated afferent fibers, also called free nerve endings, are crucial to the cessation of pain, also called nociception. What we observe here is that the receptive field of tactile receptors that span the surface of our body vary depending on the location that is being touched on our body. So we observe, for example, using this 2-point discrimination threshold task with the help of a tool called calipers, that so-called somatic acuity, so our sensitivity to tactile stimulation, is highest in our fingers and our toes, as well as on our face, compared to, let's say, the arms or the legs or the torso. So the ascending somatosensory pathway that we saw originates in the spinal cord and brainstem finally converges on the ventral posterior complex of the thalamus and reaches the somatosensory cortex in an organized fashion, which we call somatotopy. And The somatotopy has been described by Wilder Penfield and captured by his now famous homunculus. And so what we see essentially is that different areas on the somatosensory cortex receive input from different areas of our body. With respect to pain specifically, it's important to specify that pain is subjective. So there are no purely painful stimuli. Some sensory stimuli feel painful to some individuals, but do not feel painful to other individuals. So essentially, there are no sensory stimuli that feel painful to all individuals. So pain perception is supported by nociceptors, which are specialized sensory receptors. As we saw, these are free nerve endings of primary sensory neurons that are activated by noxious insults. So they can, for example, respond to extreme temperatures, in which case they can be classified as thermal nociceptors. Mechanical nociceptors are activated, for example, by intense pressure. We also have polymodal nociceptors that are activated by different types of noxious insults, such as high-intensity mechanical or chemical or thermal stimuli. And silent nociceptors are located not on the surface of our body, but in our organs, in our viscera. And these are usually insensitive to mechanical stimuli, but respond to inflammation or tissue damage. Now, here we see a depiction of the major ascending pathways that transmit nociceptive information. And interestingly, there is a phenomenon called pain asymbolia, which is a form of depersonalization for pain. Interestingly, people who display this condition experience the pain as happening to their body, but not necessarily as happening to them. And the brain area that is damaged in these cases and is part of this ascending pathway is the insula. In the case of so-called phantom limb syndrome, where people who have had an extremity amputated, for example, an arm or a hand, still report feeling pain in that missing limb. This has been thus called phantom limb. And interestingly, the brains of people who report pain following the amputation in the phantom limb and people who do not has shown an interesting type of plasticity that impacts the homunculus, the somatosensory organization. So fMRI data has shown that in those amputees that experience phantom limb pain, the cortical representation of the mouth area on the somatosensory cortex has extended into the regions of the hand and arm in these people who have had their hand and arm amputated. In those amputees who do not experience pain, The areas of the primary somatosensory and motor cortices that are activated are similar to those present in people who still have intact limbs. So this is a particular case of pain occurring in a very fascinating manner. And this is due, as we said, to the fact that the homunculus is plastic. The somatosensory cortex is plastic. It is subject to reorganization. This has also been shown experimentally in animal models. In humans, a very well-known neurologist, Ramachandran, has documented a variety of such cases of phantom limb syndrome and has also developed a mirror-based therapy to help these patients.